All right, thanks to the bitter enders here for hanging in there. We appreciate it. And thanks to my colleagues that helped with this presentation and this session. We have a good big group here that have shown interest in so-called fast processes. So my talk today is high-frequency satellite data are needed to resolve fast processes in the surface ocean. And my colleagues are Antonio. So I'm the PI of NASA's newest ocean color mission. It's not PACE, it's Glimmer. And so we intend to launch in 2027 is what's on the schedule now. And it's a hyperspectral, high-frequency spectrometer that will be in geostationary orbit. So I'm the PI. Antonio Menino is the co-PI or deputy PI. Maria Georgiou is the point of contact for applications. Blake Schaefer is on the Glimmer team from the EPA. Suzanne Craig from Goddard. Melissa Melendez from University of Hawaii. Janet Campbell, retired from UNH, and Heather Benway, who not just organized this whole event with her co-workers, but also helped a lot on this these presentations as well. Okay, so for this meeting, I want to define fast processes as those processes that exhibit significant variability at sub-daily timescales, and furthermore, they're poorly resolved by the present generation of low Earth orbit satellites. Now, I know not everyone's a satellite oceanographer in here. So there's Earth-observing satellites that orbit the poles as the Earth turns underneath them. And at very best, we can get one image a day, and because from those satellites. And because of cloud cover or operational constraints, it's usually much less than, for any given place on Earth, it's usually much less than once a day. So this presents a problem, we think, for resolving so-called fast processes. Okay, so the motivation of this entire session is we want to highlight Glimmer and its sampling and spectral capabilities. So unlike other meeting sessions, we want to enlist your help, your advice, and your ideas on what satellite products you need to support your science. And at the end, there's going to be a survey that Maria has helped out with, and there'll be, I'm a technological idiot, there'll be one of those things where you take your cell phone and look at it, and it will bring you right to it. And so if you could fill that out, that would be really helpful for this mission and folks here. We also want to gauge interest. So we have a breakout session after this where we're going to elicit your help. And we want to gauge interest in and work towards developing a manuscript highlighting fast processes. And anyone here is invited to be either submit or help be a co-author on any of that. So Suzanne Craig is taking the lead on that right now. We've got some pretty good hits on some interest in the publishing community on that. And then lastly, we want to explore opportunities and collaborations. Okay, so the outline, first of all, I want to highlight Janet's message about sampling frequency and evidence. Okay, and I'll explain what that is in a minute. And then I want to very briefly show a gallery of movies. Since we're a high-frequency sensor in geostationary orbit, we get to take the movie of the Earth, not just the snapshot every now and then. And so there has been some work done with geostationary satellites for looking at ocean processes. They're not optimized for that typically, but I'll show you some of those. And then I'll go into the Glimmer presentation. So I got a lot to do in 17 minutes. Oh, okay, so when I talk about fast processes, remember my definition. This is what we proposed to NASA for the Glimmer mission. This was kind of a gallery of processes that we were going to look at. The scientists in Glimmer are very involved, or many of them, in phytoplankton physiology and subsequent bloom evolution. So we want to look at diurnal timescales and what's happening in the physiology during the day. We also are very interested in coastal fluxes and how they affect ecosystems. And then there's a variety of applications, HAB evolution and its trajectory and fate, and then oil seeps and spills and exchanges and whatnot that all occur at fairly rapid timescales. Okay, so here's a really simple theory of why what I learned when I was a PhD student. So Janet Campbell was my advisor, and in addition to be the inspiration for Glimmer, she worked on a couple of geostationary ocean color projects that were not successful. But in addition to being the inspiration for that, before she was a professor at UNH, she had a career at Langley as a statistician. And so she brought that to all her students, and she would harp on the fact that biological processes in general, rates and even stocks, and certainly episodic processes, are not normally distributed in time and usually not in space. And so all the assumptions that we make about the normal curve and sampling from that and the behavior of that, a lot of times I give it credence in papers, might not be normal, or I did this, test on it and it was kind of normal. But all the assumptions that we make on that go out the window in reality. And so I've taken a data set from UNH, from a buoy that's been out there for 17 or 18 years. They do a good job taking care of it. It's NOAA funded. And we measure fluorescence on it every 3 hours. And I'm showing the distribution of 200,000 plus samples from that. You can see that it's not normally distributed. And if I transform it in log space, you can see that it's fairly normally distributed in log space down here below. Okay, so if we take that, distribution and model a log normal curve to it, it fits very nicely. And we see that the mean of those data were 1.89 milligrams per cubic meter, but the median was far less than that, half that virtually, and 1.01, and then the mode is a fraction of that. But here's the thing: if we are doing independent sampling of that chlorophyll over time, okay, what's the most likely value that we're going to grab at any given time? It's the mode. It's certainly this part of the curve right here that's most frequent. Okay, so Janet's simple message, that unfortunately not even I pay attention to all the time, is that undersampling of a log normal distribution commonly leads to underestimation of the mean or even the median. And keep in mind, over any given time scale, we usually want the mean behavior. If for primary productivity, we want, the mean, might be a weekly mean or it might even be a daily mean, but we want the mean. And yet those processes, that primary productivity is log normally distributed. So we have to be able to get a method that will sample at the native time scale of the process, okay? All right, and so I took those data, just to give you an example, here's the evidence. And these are fluorescence data, and probably Yale is going to come up in the next talk and tell me why I shouldn't have done this. But these are fluorescence data, and you can see that the mean here is 1.89. And what these numbers are right here are a sampling simulation where I grabbed all the data at one day during the Modus overpass around 1:30 p.m. I did it at one day, and then every 3 days, and then every 5 days, and then every 7 days. And you can see that there's a significant underestimation of those means from those overpass times. And so, it's anywhere between 14% and 24%. So that's an example. Might not be the best example, but conceptually you get where I'm going. Okay, so Zongping Li did a nice numerical simulation with average daily PAR and chlorophyll. And so what he wanted to show are differences if we had a geostationary sensor collecting PAR and chlorophyll throughout the day versus just using that daily mean, which is what we do right now. Okay, so primary productivity algorithms, which have totally changed our understanding of global production from space, are based on averages, sometimes monthly averages, average PAR and average chlorophyll. Okay, and so what he did then was to use a average over the course of a day, and he got that blue line right there as the mean for daily primary productivity. And so then he took a realistic a PAR and a realistic time varying chlorophyll or biomass, and he did the simulation there, and he gets that curve. When you integrate that curve, or get the mean from that curve rather, you get that line that's below. And he had this awesome, which is underestimated. He had this really nice line in there that made me pay attention. Without consideration of the coupling of diurnal variations of both biomass and PAR, daily primary productivity could be overestimated by 52%. 52%, you know? So everybody, I always say, you know, the coastal zones are the most productive in the world, even though they're 10% of the area or 7% of the area, they contribute to X productivity. And so, one could argue that we need to examine exactly what that number is. Okay, so a group of us worked on a paper a couple years ago where we were trying to use the GOCHI, the geostationary ocean color instrument from Korea, to actually look at hourly changes in chlorophyll. Actually, what we wanted to get at growth is chlorophyll growth rate, or phytoplankton growth rate. We didn't quite get there, but the paper got published anyway. And so, so what you're looking at here in this figure is every color is a difference in chlorophyll over the course of the day. Okay, 9:30 minus 8:30, 10:30 minus 9:30, et cetera, et cetera. So that's what the colors represent. And we did this over a whole year in a region that's in the EC between Japan and Korea. And it was a highly productive, it was a productive region, but it was case one water. So that was our criterion. We wanted to get clean water that was only influenced by phytoplankton. And so, so what you can see here over the course of the year 2016, there's a mean curve there in black, and none of those hourly differences look like the mean curve. So that's interesting to us. So, implications there, if you were coming over at 10:30 every day, or if you were coming over at 1:30 every day, you'd have very different values, and you'd make up very different statistics over the course of a year from that type of sampling. So that's a point. So we see it in the satellite data. Okay, I want to move over to a couple of movies. I have more. Our colleagues from Korea have fed us several of them from GOCHI. GOCHI's been a total inspiration for getting Glimmer and OCX, which will be a following ocean color mission from that's geostationary from NOAA. And then also our friend Ryan Vandermeulen at NOAA supplied one of these. But this just gives you a flavor of what we can see with geostationary data versus polar orbiting data. So polar orbiting data, snapshot. Okay, so this first one, whoops, let me back up here. Oh dear. Yeah, this first one, I don't know how well you can see this, but I want to call your attention. This is northeastern Texas right here after a storm. These are sediment plumes, these red blotches right here. And this is over the course of 6 or 7 hours using a sensor, a meteorological sensor. This is the sensor that you use when you see the weather patterns, track across the United States on the news at night. So this is an ABI, which has a visible band or two, and you can see sediment coming out of those. And from my eyeball, when I looked at this versus a Google map, you know, it seems to be about 10-kilometer movement in a lot of these plumes over the course of a day. That's a lot. That's a fast process. Okay, and so this, again, this is not optimized for ocean color at all, but it shows visually what's going on. Okay, so here's another sediment plume, and this is influenced by tides here, and this is I think somewhere around here is Seoul, South Korea, and this is sediment from resuspension, from tidal resuspension. And I just wanted you to check out the beautiful tidal ellipse that they got over 6 hours with the Gochi satellite there. You see that elliptical pattern one more time? Okay, so this fast process, you know, imagine we only had one image. This is ice forming over the course of a week or so up in the Bohai Sea, up of China, in northwest of the Yellow Sea. Let me show that one more time. But you know, here's a relatively fast process. It's kind of a novel application for ocean color data, but it you know, good for navigation and whatnot. And sometimes this freezes over really, really quickly, even faster than that. And then here's a real novel application. Keep in mind, these are 500-meter data here, and the Koreans used this to track a ship, or they found this ship that was dumping something. And so this ship was following a pattern like this, and you know, they could estimate its, so check that out one more time. They could estimate its speed from the satellite imagery. So this is over the course of a day, and they estimated, and so so that was really interesting. And you know, there'll be a lot of applications that we'll come up with by using ocean color, hyperspectral ocean color data to look at the Earth. Okay, so now what are we up to in terms of Glimmer? So this is a presentation that I hacked from Antonio Menino. We typically share slides, and we always have contributions from the Glimmer team. So we don't do this alone. Okay, so Glimmer is the geostationary littoral imaging and monitoring radiometer, and we call this observing coastal processes from space at an hourly frequency. And lots of acknowledgments here, but it's being led out of UNH. Goddard is an important partner for data processing and science. It's being built by Raytheon Intelligence, and well, Raytheon's changed their name twice since Glimmer started, so it's Raytheon something out in El Segundo, and then Southwest Research Institute is doing mission assurance and some engineering. Okay, so a glimpse into Glimmer. Glimmer has these wonderful, you know, it kind of hits a sweet spot on a lot of sampling here. It's going to be hyperspectral, similar to PACE, although PACE has SWIR bands at all, but from the UV up into the NIR. We have a prism, so what that means is we have a dispersion of light across the spectrum. So we have this awesome spectral sampling down in the UV and the blue, and then it gets a little bit wider, up to 7 nanometers in the red. But anyway, think about that, what you can do, what kind of gases or whatnot we might be able to retrieve using this really high spectral sampling. It's got, you know, our selling point is the high temporal resolution. We get hourly scans of the Gulf of Mexico. We get 2 to 3 times a day everywhere else in the continental U.S. And that's about as good as OCX, you know, a very expensive project from NOAA that launches in the 2030s, early 2030s sometime, is going to be able to do. And so, and then we have 3 times a day in other regions. We're very interested in tracking HABs, so we can move this gimbal around and track things with it as well. We've got decent spatial resolution, 300 meter at nadir. This is the ground sampling distance. The pixel will be a bit bigger than that. And we have very good high signal to noise requirements. But the thing that's very exciting about Glimmer is we have this adaptive scheduling and targeting such that if you, the community, call or get on the web or however we figure out how to do this when we launch and tell me there is this amazing, or tell us, this is amazing bloom, or hey, there's an oil spill over here, or there's something going on over here that I really want you to look at, we can spin around. It will take us a day to do this, but we can spin around and we can stare at that at very high temporal frequency. You know, maybe if it's a small area, we could do it, you know, every half hour or so. So that's pretty exciting, and I hope we get to use it, and more about that later. Okay, so I did want to make a point. This is a thesis work from Greet Nukermans. This slide's a little bit out of place, but the idea here is that we're going to have a lot more data in general because we're imaging, you know, when it's not cloudy. So here's a sediment map that Greet made using a meteorological sensor called Severi. And she aggregated these and averaged these over the whole day. And here's the nice coverage you have. And here's the single MODIS image the same day. And, you know, we've done estimates in the Gulf of Mexico. We're going to probably have like 30 more, I'm giving myself 2 more minutes, 30% more, you guys can't do that, 30% more, I'm sorry, 60% more data. So our science is focused on phytoplankton growth and physiology and short-term coastal processes, and so how one affects the other as well. And so we have a applied science, several foci that you know, we did not propose a big applications team. We gave a little tiny bit of money to a overworked, workaholic, underfunded Maria Georgiou, who has really grown the applications efforts exponentially with very little support from us. And so there's been this organic outpouring of interest in these types of measurements. Once people start thinking, oh, pace every hour, then they call her. And so we are more on that later, but this is an area that we've had a lot of interest in. Okay, just our daily schedule is, so we're not $1 billion mission, so we have to kind of target areas, so Gulf of Mexico, six times a day at least, more in the summer, maybe less in the winter. CONUS 2 to 3 times a day, very interested in the Orinoco and parts of the Caribbean. We can do this adaptive scheduling to find sargassum and whatnot, and we have starting to make plans for that. And the Amazon, we're really interested. But we also come over here and look at these blue-violet waters for calibration purposes, and we match up with PACE several times a day, like 5 or 6 times a day, and that's for calibration purposes. Okay, and I just have 2 more slides, and this is a simulation of our scanning efforts from 95 West. We don't know exactly where we're going to be, but Adrian, see how close we are to the Galapagos there? And we just need to look down. And so you see we follow the sun. Here comes night. So we're moving over to the west a little bit, and we're going to go down the Pacific there before night gets us, and then we start again. So it's a pretty good operational plan. And my last slide, I just want to show you or mention that I want to mention that this is the same data processing that all the NASA satellites go through at Goddard, so we feel good about that. We have a list of ocean products here. We've ripped all of these off from PACE. Thank you very much, PACE team. We're really leveraging PACE. But then we have some of our own too that are, some of them indeed are dependent on geostationary high frequency data. And that's it. And so we welcome advice, comments, and ideas for collaboration. So I was 4 minutes over, so I apologize for that.
So while the slides are coming up, hello everyone. My name is Yaila Sezginer. I'm a PhD student at the University of British Columbia. I work in Philippe Tortel's lab, where I study phytoplankton photophysiology and productivity using variable chlorophyll fluorescence methods. So hopefully you'll forgive me, but this cover slide is actually a little bit misleading. All of my data comes from underway shipboard measurements, but in this talk, because of the session, I was inspired to try and relate the underway measurements that I have to the satellite systems and what we can learn from something like a geostationary satellite like Glimmer. So as we heard in all of the talks on Tuesday, phytoplankton play this really outsized role in shaping the chemical landscape of our oceans and our atmosphere through photosynthesis. And their sinking rates, their elemental composition, their growth rates, metabolic rates, all of these things are highly sensitive to the nutrient availability in the water that they grow in, as well as the micronutrient availability. So I added those after hearing the talks on Tuesday. But the point that I'm making here is that there's this tight coupling, these feedbacks between biogeochemistry and phytoplankton, and those feedbacks are really mediated by phytoplankton physiology. I focus in particular on photophysiology, which begins, of course, with the photosynthesis light reaction. So here we are at the start and heart of it all on the thylakoid membrane in a chloroplast. So we're looking at the light reactions, and this, yeah, nice, okay. And so photosynthesis is really initiated when the reaction center within photosystem 2 absorbs incoming light energy. It becomes energetically excited, and it needs to return to ground state, which it can do either by releasing an electron, which is accepted by the primary electron acceptor molecule QA. It will then travel through the electron transport chain and ultimately provide the reducing power needed for downstream carbon fixation. Alternatively, photosystem 2 can return to ground state by releasing energy as chlorophyll fluorescence or as heat. And because these are competitive pathways, how much fluorescence we get is inversely proportional to the photochemistry. And the photochemistry pathway really depends on whether or not there's an oxidized QA molecule ready to accept an electron from the reaction center. So I like to use this analogy from I Love Lucy to explain this, if there's any fans. Lucy and Ethel are photosystem 2 complexes, and their job is to take incoming light energy and convert it to chemical energy, wrapped up chocolates. Yeah, they can only hold one chocolate at a time. They can only wrap one chocolate at a time. Similarly, the QA molecule can only hold one electron at a time. And so when the chocolates are coming out slower than the reoxidation rate of QA, they're doing great. They're very efficient. And when those chocolates come out faster and faster and faster, they start to get inefficient. The chocolate energy has to find alternative pathways to go down. And so very similarly with photosynthesis, when QA is oxidized, we have very efficient photochemistry pathway. But when QA is reduced, it's already holding an electron, the photochemistry pathway is closed, and so we see an increase in fluorescence. So variable fluorescence techniques operate by manipulating the redox state of QA. And so I work particularly with fast repetition rate fluorometry, or FRF, and this operates by first supplying a really rapid excitation sequence where these pulses of excitation light far exceed the reoxidation rate of QA. This forces an increase in the fluorescence until fluorescence stabilizes at the maximum level when the photochemistry pathway is closed. The QA pool is all reduced. Then you supply a relaxation sequence where you have an exponentially increasing interval between those pulses, and that allows fluorescence to return back to basal levels as QA is reoxidizing. This curve tells us a lot about the photophysiology of our samples. So you can fit a biophysical model to this described by Colbert et al. In 1998 to get the functional absorption area of photosystem 2, or sigma PS2, over here. And there's the reoxidation rate of QA, or tau QA. And then there's a very commonly cited photophysiological parameter called FV over FM. That's the photochemical yield of photosystem 2, and that's really the amplitude of this curve. And if you're interested in getting involved with these instruments and don't know where to start, I would really point you to Schubach et al. 2021. That's a great paper where members of the community synthesized this kind of results or methods and also provided recommendations. So Really, the beauty of the fast repetition rate fluorometry method is that you can make those fluorescence transients in about a second, and this offers really great opportunity for high spatial resolution, temporal resolution, high frequency sampling. So this is an example from the Southern Ocean from a cruise I was on in Christmas, and you can see FV over FM is quite low in Drake Passage, where we know phytoplankton are iron limited. But then every time we cross one of these hydrographic fronts, there's this big blip in FV over FM, presumably because of mixing and reintroduction of nutrients to the surface. And this is the kind of thing that would be really hard to capture with discrete sampling. It'd be really hard to line your stations up with those hydrographic fronts that move in space and time. So hopefully that's fairly compelling, but now I'll give you a caveat. When you're thinking about using FV over FM or fluorescence methods as an indicator of nutrient stress, you have to be really careful to think about the other things that affect fluorescence, especially the light environment. They're very sensitive to light. Particularly, there's this photoprotective mechanism called non-photochemical quenching. And this is an increase in the heat dissipation. So you get a decrease in fluorescence that's not due to photochemistry, hence non-photochemical quenching, or NPQ. This is triggered when you have excess excitation energy that causes the charge separation at photosystem 2 to far outpace downstream photosystem 1 and proton pumping at ATP synthase. And so you get a buildup of acidic conditions in the lumen, and that will eventually reach a pH threshold that causes a conformational change in photosystem 2 that promotes heat dissipation. And so this is what your fluorescence signal will look like when you have non-photochemical quenching. In the dark, you have this larger curve, and then when you have ambient light, that curve becomes extremely compressed. And of course, that will affect all of those photophysiological parameters you're deriving from the curve shape. And in a diel or in a time series data, you get these diel cycles because of it, where you have an increase in the photochemical yield, or FV over FM, overnight, and then that sharply decreases during the day when NPQ is active, and similar with the functional absorption area of photosystem 2. So again, if you're trying to use fluorescence methods as an indicator of nutrient stress, it's really important to account for these non-photochemical quenching mechanisms. And the most common way people do that is they just take their samples and they hold them in the dark for a long enough time that they think non-photochemical quenching has relaxed. However, I would argue that non-photochemical quenching itself is a really useful photophysiological parameter. In 2020 and 2021, there were a bunch of papers coming out using it as an indicator of iron stress, particularly in the Southern Ocean. And then in 2015, a former student in the lab that I'm currently working in, Nina Schubach, found this really strong correlation between non-photochemical quenching and the electron requirement for carbon fixation. This has really great practical implications because, as we've shown, you can get a very high resolution FRF sampling and estimates of electron transport rates, whereas measuring carbon fixation is more challenging. It requires discrete measurements most of the time. And also, you're using radiocarbon a lot of the time, which is a lot of paperwork and labor intensive. So this offers a lot of potential. And it might seem like a somewhat arbitrary correlation at first, but if we go back to what we know that non-photochemical quenching is a photoprotective mechanism for diverting excess energy. And there's all these other mechanisms for diverting excess energy downstream of photosystem 2 as well. These are alternative electron transports or cyclic electron transports, and they really act to maintain redox homeostasis when you're getting charge separation over here that outpaces photosystem 1 activity and ATP synthase activity. And this will show up as an increased electron requirement for carbon fixation. So when we think of these things together, then it starts to make sense why we see this correlation between NPQ and electron requirement for carbon fixation. So now I have absolutely word vomited everything that I've learned in 4 years at all of you, and hopefully everyone's doing great. And now I'm going to try and transfer all of that knowledge over to the satellite world. And I should say that I'm very new to the remote sensing. I pretty much was inspired by this meeting to try and learn more about it. So if I say anything that's completely wild, feel free to correct me. So FV over FM, or those FRF measurements, are extremely comparable to the satellite product fluorescence line height, which when you divide by chlorophyll gives you fluorescence yield. So this is an image from Barenfeld et al. Of the global fluorescence yield. And we actually heard quite a few talks on Tuesday talking about using this parameter as an indicator of iron stress. And now I'm just going to lay out the different kinds of stresses that we see in the ocean and how these affect both types of fluorescence methods or fluorescence results. So in the subtropical gyres, we often have macronutrient limitation. This affects protein synthesis rates, but because these are biomass normalized measurements, they actually don't cause a really big signal in the fluorescence data. We don't see a huge compression of the FRF curve, and similarly, the fluorescence line height is pretty just, I don't know, close to the mean value there. Then with iron limitation, you get a really significant compression of this FRF curve, and you get a large signal in fluorescence line height. And I should say fluorescence yield, I'm sorry. But that's partially because that under iron limitation, you have an increased photosystem 2 to photosystem 1 ratio, because photosystem 1 is very iron rich. And the fluorescence that we're measuring is almost exclusively coming from photosystem 2. So that's why we see this increase in the fluorescence line height measurement. And then lastly, photoinhibition is also a biggie, and this causes a big compression in FRF curves as well that look very similar to the iron limitation. And what do they do to the satellite data? I'm honestly not super sure. I haven't seen in the literature a lot of evidence of what it does to the satellite data, and that's partially because of the way that the satellite data is measured. So as we heard, most of our ocean observing platforms are sun synchronous or polar orbiters, and this means that the measurements that they're taking are always coinciding pretty much with midday when the sunlight is at its peak. So we don't have low light measurements to compare against, so we can't really say actually what the photoinhibition state is. So we still have that problem that we have with the FRF data is if you want to use these as indicators of nutrient stress, you have to be accounting for the non-photochemical quenching. And how do you do that when you can't hold your sample in the dark and wait for non-photochemical quenching to relax? So Barenfeld et al., which is really the paper that described these measurements for the first time and what you can do with them, they applied this correction factor that's an inverse relationship of PAR scaled by the mean global PAR. And you can see that it explains the data fairly well, and also we can repeat it with the FRF data, and we get a similar curve. So that's helpful. But there is quite a lot of spread around that curve, presumably due to photoacclimation. So maybe that mean global PAR isn't the right photoacclimation state for all phytoplankton coming from different latitudes, different seasons, et cetera. And so there's a theoretical framework that phytoplankton acclimated to different light levels should follow along different curves. They should be scaled by different light measurements. And again, we can repeat this with the FRF data. So here is measurements made at night, and here is measurements that are made during the day when phytoplankton are acclimated to higher light levels. And we can see that it follows the theoretical framework quite well. So that's a good thing. And then now is where I start to get into the kind of speculative area where I'm starting to ask questions that I don't know the answer to. But can we use geosynchronous satellites to kind of fill in a lot of those data gaps? So as we heard, we have a new geostationary satellite coming out soon, Glimmer. So this is really the time for us to voice our thoughts, what we want to see, the kinds of questions we want to ask of Glimmer. And at present, I think we only have one geostationary satellite that's designed for ocean observations, and that's GOKI. And from what I understand, this satellite doesn't have a PAR product. And that means that when people in the past have tried to use this geostationary satellite to constrain non-photochemical quenching, again, they found that these different curves do help better describe non-photochemical quenching effects on phytoplankton that are acclimated to different light levels based on the different seasons when they were sampled during. But you can see the data is still quite sparse. And from what I understand, that's because they had to line up their measurements with MODIS to get that PAR product. So hopefully with Glimmer, we can really fill this in more and get a better understanding and a better idea of the photoacclimation and non-photochemical quenching terms. And then this is where I really go off the deep end and get super speculative. So yeah, you can come check me later. But I'm wondering if we can collect some sort of electron transport rate measurement from the geostationary satellites based on fluorescence yield measurements, very similar to how we do with fast repetition rate fluorometry. And if we can then apply this correlation that Nina found to get a physiology-informed, satellite-based gross primary productivity algorithm. So that could look something like this. This is speculative. And that's kind of where I'm left at. And so now I'll wrap up by summarizing everything we went through because it was quite a lot. So we started in the first half with talking about phytoplankton photophysiology and how it can be rapidly measured in situ with chlorophyll fluorescence techniques, and how that can be a powerful indicator of nutrient stress if and only if non-photochemical quenching and photoacclimation are accounted for. Then we moved on to talking about geosynchronous satellite potential and how we could try to collect non-photochemical quenching estimates over more complete day-night cycles. I'll note we heard that the GOSI will have 6 measurements per day, and also I don't think that we could get night measurements actually because you need some light to induce fluorescence in the first place, and we're relying on the sun in this case. So yeah, and I think at very low light levels, we would have trouble with signal to noise ratios. And then, anyway, moving on, hopefully we can still use what we have with those 6 daily measurements to get better photoacclimation and non-photochemical quenching correction factors. And then maybe we can leverage all of this information to develop a physiology-informed gross primary productivity algorithm. So with that, I'd like to say thank you all so much. Here's my contact information, and I'm hanging around here until tomorrow, so you can come find me and ask questions. Thank you.
We have time for a question if anybody wants to ask one before we transition to the next speaker.
Yeah, that was an outstanding presentation. I just want to mention, I believe there is a PAR product for GOCI. We used it in 2021, but it wasn't available to O'Malley in 2014.
And if there are any Korean colleagues here, there are 2 GOCIs.
All right, so my name is Melissa Melendez. I'm research faculty at the University of Hawaii. I'm just going to start really quick. Today I'm going to be talking a little bit more about CO2 carbon chemistry dynamics at IUNA timescales on these buoys that are located in sensitive areas like coral reef systems. These buoys have been there for already almost more than 10 years, and they are supported by the NOAA Ocean Acidification Program and the Pacific Marine Environmental Laboratories, and a lot of colleagues here that help maintain these buoys for many, many years, and also work with the quality of the data collected. So I already we already worked during my PhD with Joe on the methods on how to analyze this data and how to understand a little bit more about the PCO2 and diesel inorganic carbon drivers at different timescales, but we focused during my PhD during seasonal dynamics. And right now what we want to do is to kind of synthesize all of this data and understand sub-daily kind of processes. So I hope to apply some of the methods already developed. And here, just to give you an overview of the PCO2 variability across all of these stations. And here, just the message is that I want to see, similarities and differences between different ocean basins. And here you can see Pacific buoys and the Atlantic buoys. And the message too here is that we see large gradients compared to the oligotrophic waters where these buoys are surrounded by. And also the dots are data that are most extreme data that are no outliers. So this variability is related to the diurnal processes. And what I wanted to see was to see the distribution or the variance across different frequencies. So what I did was that I changed my time domain to a frequency domain using spectral analysis to try to identify those diurnal variabilities and frequencies. And here we can see, this is in PCO2, and we can see significant frequencies coming out at the diurnal cycles, 24 hours to 25 hours, semi-diurnal, 15 to 12 hours, and then we have a significant frequency too at 8 hours, which could be related to the tidal component at some coastal areas. This is not in all of the stations, and it's not in all of the years. It's very interesting to kind of like now, this is an average of all of the years, but it would be nice to see like every year how these frequencies change. And I also want to point out that with this data and talking about frequency of your data collected, we take measurements every 3 hours, and the processes that we can understand are processes that are higher than 6 hours. Processes that happen less than 6 hours, we cannot really understand because then there are errors coming out. And this is known as the aliasing frequency or aliasing errors. We can talk a little bit about that later. But I wanted to see then how this diurnal frequency variability is changing over time. And this is just the peak-to-peak diurnal variability averaged by month during the whole years for all of the stations. And we can see that these amplitudes are actually changing over time, and they are different between stations. And we can see also that some of them are increasing, like the one in Hawaii, and some others are not increasing, but some of the years are, these amplitudes are like muted, and some of them are amplified. So this is how satellite data can maybe help us understand how regional processes are affecting these coastal areas. And this is an example of the one located south of Puerto Rico, in the south coast of Puerto Rico, the Caribbean Sea. And we can see in the first image chlorophyll data. The higher values are in green. And then the second one is the salinity with the low salinity in blue. And in this particular event, we see the PCO2 coming down for 150 microatmosphere, and the diurnal variability was muted for more than 10 days. So I'm just going to leave it there. Thanks.
I'm Nathan Hall, UNC Chapel Hill. I'm going to talk a little bit about how phytoplankton overcome this ubiquitous problem of vertically separated light and nutrient resources. I'm going to start off talking about estuaries, then at the end, talk a little bit about the possibility that they're using some of these same strategies to solve this problem in the open ocean. So this is me. The first estuaries I'm going to talk about are the Neuse River estuary. Dumps into Pamlico Sound. It's a eutrophic estuary, lots of algal bloom problems, lots of interest in trying to figure out what's regulating the community composition. Even though it's eutrophic, it still has this issue, except for the very head of the estuary where the river dumps in, there's almost no nutrients in the surface water and no light once you get down a little bit into the water column. So a lot of this work I used on these vertically profiling buoys that would cast a YSI sign with a fluorometer on them. And so I could see temperature, salinity, and where the chlorophyll out was in the water column. And these would, every 30 minutes or so. And we did this for a couple years. We have a lot of data. A long time ago, too. That was me back when I was a little bit younger. This is the kind of data that we got. So, it's about a 6-meter deep water column. The bars across the top shows the day and night cycle. White's day, black's night. The phytoplankton, these are flagellates, and they're just swimming up during the day and down at night as regular a pattern as the tide. This is diel vertical migration. Yeah, so you can just see big clumps of red up near the surface during the day, big clumps of reddish yellow down in the bottom water at night. That black line drawn through that, that's just the depth of the fluorescent maximum. That becomes important to this next slide, which shows how ubiquitous of a pattern this is. So this is a wavelet power spectra of the depth of the fluorescence maximum over a 1.5 year period. And you see the big red bar is the cycle 1 per day. That's a diel vertical migration signal. This process happens every day. And so the black and white is across now as data gaps, black are data gaps. So you see really where there's not power at 1 per day, it's because we didn't have any data. So this is an omnipresent problem, and at least in this estuary, it's an omnipresent solution. I didn't just use these buoys, I also spent some time sitting in a boat out making measurements. This just shows an example of 2 days, one was a cloudy day and one was a sunny day. It was in June, July. You can see that there was a lot more light in July than there was in June. On the day where there wasn't any light, the phytoplankton just swam straight to the surface. And they hung out right at the surface. And the net effect on primary productivity measured by C14 incubations at different depths in the water was that migration to the surface compensated for the lack of light. You can see the panel C, there's almost no difference in those productivity profiles. So this is, these short-term migration patterns can be huge for access to light and for optimizing light availability. One of the cool things I saw with those profiling buoys too was this pattern of a lot of times the phytoplankton would swim up to the surface, they'd get there around 9 o'clock, and then, oh man, it's too bright. And they would go down at lunch, and then they would come right back up in the afternoon before going all the way down again at night. So those kind of patterns, I started thinking about for this session, we might be able to see those using something like Glimmer, these short-term processes. But if we don't know what they are, it can also really make interpreting products that Glimmer puts out really hard. I did a little bit of work trying to figure out how much these kind of patterns might increase productivity of estuaries. Turns out not huge, maybe 5 to 10%. These patterns are also important because they minimize flushing losses. So we have 2 layers, ups, part of the water column is going downstream, and then bottom water is coming into the estuary. So if you split time between layers, you decrease your net advective losses. That's been shown in other estuaries as well. And, oh, crud. It goes so fast. All right, so I'm just going to skip it. They take up a lot of nutrients in the bottom water. The coolest thing, though, is this new set of papers coming out of Germany, where the idea is that in the deep ocean, they can't possibly do this in a day. But a lot of the nanoflagellate community and a lot of the bigger diatom stuff are probably doing this over the time scales of a week. Growing slow, moving down over tens of meters, down to the nutricline, back up to the deep chlorophyll max. This process may be really important. It hasn't been proven, but there is some observational support. I encourage you to read these papers, the Wirtz and Smith paper and the Wirtz et al. 2022 paper. Big implications of this. If it's true, this migration may be responsible for about half of the MPP in the ocean, and it drastically changes the outcomes with what might be happening with climate change. Apologize for that.
Good morning, everyone. Our next speaker is Chris Osborne from North Carolina State University.
All right. Hello. Nice to see everyone. And another Carolinians up here going to tell you about what's going on in the Neuse River Estuary and in the Pamlico Sound. So as the title suggests, we're going to talk about the downswing of the seesaw. So you're going to learn what the seesaw is. And we're going to think about how extreme weather events, such as hurricanes, accelerate the loss of carbon from the landscape into estuaries and then ultimately into the coastal ocean. So I'd like to acknowledge the incredible team of students, postdocs, and collaborators I've had the good fortune to work on this problem with over the last several years. So the aim of the talk is to look at carbon loss from the landscape. We're going to look at it as dissolved organic matter. What is the seesaw? Well, here's a little schematic to show what this is. It's actually an acronym for carbon saturation and weather. It started thanks to OCB as a working group and workshop with some other sponsors we were able to have in Raleigh in 2022 and then some subsequent meetings over the last couple of years here. And the idea is that you have a buildup or a saturation of carbon in the landscape, and then at some scales, temporal scales, we don't have really well worked out yet, that carbon gets transported into the coastal ocean through estuaries, these reactors where some transformations might take place. Other time scales that we don't really have good handle on, nor how these these time scales are changing with more extreme events, more heavier precipitation, these types of things. So how fast is the seesaw happening is something of our scientific interest for this working group, and Glimmer is hopefully going to be a fantastic way for us to try to look at that. For the talk today, I'm going to focus on one part of dissolved organic matter, the colored or chromophoric part, the part that absorbs light, we call CDOM. So it's a key reactant in the coastal carbon cycle. You can see here on the, your left, that in that cartoon, that there's a number of different sources for it. We're going to talk a little bit about how sources of DOM change, how sources of change as a function of where it's coming from in the landscape. And then just think a little bit about what are some of the outcomes. So we have these fast processes that we might be moving carbon into the coastal ocean, but what does that mean once it gets there, when it gets into these reactors and perhaps sits around for a good period of time where it can be worked upon. And so we can think about this as the seesaw as some manifestation about how these reaction rates, how resonance time in different estuaries and coastal systems affect processes, and as well, looking at these hot moments and hot spots, so where these extreme episodic events are occurring. So it turns out that North Carolina, like South Florida, and you may have seen in the news, South Florida got inundated with an extreme event just recently, just yesterday. I think it was nearly 17 inches of rain, 24 hours. We're talking about really extreme events. And that's due to climate change, of course, and it's also manifesting as extreme flows in our fluvial systems, our rivers, and leading into our estuaries here. So North Carolina, coastal North Carolina is a great place to sort of look at this problem. And as well, it's very fortuitous that I'm able to collaborate with folks like Nathan and Hans Pearl and others for these observational programs that look along the main axis of the Neuse River estuary that you can see here. We're going to focus on the tidal freshwater, the mid-estuary, and the lower estuary into the Pamlico Sound. And then from NC State side, where Raleigh can't quite see there, it would be kind of behind Station 7 or so. But we look along the river and kind of connect with this program and do some observations on how the river's changing as well. The other thing I want to point out from the image on the right is that where you see these green markers, this is where the river encounters an enormous amount of wetlands in that environment. And what we're understanding is that those end up becoming very important for the quality of carbon that makes its way into this system and can potentially be reacted. The other thing I'll point out too from this little table is that the freshwater flushing time, so the resonance time, can vary quite a bit. So what comes into that system can hang around for quite some time and be transformed. So what does the Neuse River look like from space? Well, here's a beautiful image in 2018 of the different colors that you can see here. Here's the CDOM filling up the Neuse River estuary. But then I also want to point out, look what happens, or look along the coastline and these sounds here, these lower Outer Banks sounds, you can see there that that brown isn't so present, and then of course the coastal waters are nice and blue. What happens when something like this occurs? This is Hurricane Florence in 2018. We got about 15 inches of rain across the Neuse Basin, and this is how the image changes. You can see the brown still abundant and flowing into the Pamlico Sound. You can also see sediment as well as these plumes of CDOM coming out of the inlets into the coastal ocean. So these extreme events can very rapidly move that material from the landscape into coastal waters. That's what the seesaw is in part focusing on. So we started looking at how CDOM concentration can inform on this. One of the things we see is that there's a pretty good relationship between the amount of CDOM measured as an absorption coefficient at 350 nanometers and the concentration of wetlands in the watershed. Pretty sensible stuff, happens all over the place. On the right here, what we're looking at is a plot of, this is a stable isotope concentration of dissolved organic carbon plotted against the SUVA value. This is a CDOM product. It's just the absorption coefficient at 250 nanometers normalized to the DOC concentration. So in this data space, what you can see are a bunch of green and yellow dots. Those are observations of the Neuse River estuary and Pamlico Sound after Hurricane Matthew in 2016. And what we saw there is it kind of fits together with this story about the increase in the CDOM concentration and where it's coming from. It's not just coming necessarily entirely from the landscape upriver, but it's coming a lot from these wetlands. And I point that out as these dots being bounded between the regions for the upland river and then the wetlands in that transition part. So that kind of gives us this sense of how we wanted to go back, and what I've been working on lately is trying to see the seesaw in some of these observational data that we have from this monitoring program. So we're going to take a multi-year view where we have observations of absorption and concentration, and then we're going to shift over and focus on fluorescence. So we're coming back to that phenomenon of fluorescence we just heard about. So these plots are going to show, again, the observations here right at the estuary mouth, and this is CDOM concentration over that time span. The middle graph, this is antecedent precipitation for 7 days before the sampling event. So right there, you can see that we're getting into that variability and trying to understand how these processes are occurring in terms of these fast processes and what Glimmer will be able to tell us. And then in the bottom graph is the discharge of the river measured just above head of tide here. And you can see that where we have these large flows driven by these events, we can see spikes in in CDOM concentration, also in DOC concentration. So we're seeing it in both of those. And the one thing we wanted to do next is to try to unpack where that carbon's coming from. And we can do that using fluorescence. We can use CDOM fluorescence to basically fingerprint these different sources. And then we have a modeling approach we use to do source apportionment there. It kind of looks like this. This is what the fingerprints look like for the upland river, for a soil leachate from soils within the watershed, and then from the wetlands that are adjacent to the river and near the estuary. And so what we do, we call these EAMs for excitation emission matrices, and some of you in the audience may have heard of a statistical technique called parafac. We don't do parafac at all. It's fine for doing some things, but what we found, and I say we, I should also acknowledge my colleagues here who are statisticians at Duke that can actually do some pretty amazing and rather kind of parsimonious ways of doing statistical models. they helped me develop this regression model. So the first thing you do is you create this dictionary of these sources. You got to kind of go into this knowing what your sources are. And then it's a non-negative least squared regression approach where we can take and build a dictionary and then very accurately resolve those sources using these EANs. If you're interested in hearing more about how that works, be happy to talk about that. It has a lot of different applications. So what we did then is we wanted to take this approach and think about this kind of experiment saying like, okay, we have this fast event. We're going to saturate the interface along the land to ocean interface here with a rainstorm. So we'll look at a couple of different events and then focus in on Hurricane Florence. But the idea here is that we've got when there's no storm, you've just got the river sort of flowing through this wetland area, and it's disconnected from the wetlands. The flooding that occurs then connects these wetlands, and so the seesaw is operating to sort of undersaturate the landscape by moving the carbon out, but then taking and oversaturating the receiving waters with that translocated carbon. So that's kind of the idea here. So the first thing we'll look at is a plot of these three different sources from the RTS fluorescence model, the river, the soil, and the wetland. And then we're looking at the tidal freshwater over that same time series that we examined before. And so one of the things that you can see is that at the tidal freshwater reach, just below where the wetlands are there, you can see it's generally dominated by those wetland signals. There's also some soil sources and some river sources that we're estimating. So that makes sense. Okay, we can kind of see some variability in that happening. But then thinking about how these fast processes might translate into different time scales for how long the material sits there and then ultimately what happens to it, take a look at the same sort of plot, but now mid-estuary. You can see that these fractions persist as they move down the system. And if you think back to those satellite images I showed you, it makes sense. The river's pretty brown, or the estuary is pretty brown. But even down as we get into the sound and thinking about reaction in the Pamlico Sound proper, as well as the export through these narrow passes, you can see that the DOM maintains the dominance of that terrestrial signal, certainly, but very prominently these wetland sources. So that's kind of how it looks at this multi-year time scale. If we focus in on 2018, we see the same kind of thing happening, and especially the effect of the extreme event of Hurricane Florence. Let's run through that. These are just different days throughout 2018, right before the storm hits. So the storm comes through in between September 4th. The next sampling is September 26th. And you can see here that shift. Storm comes through, shifts the sources. We're just looking at the fraction of the wetland now. But again, that wetland signal persists and stays strong, probably supported by subsequent, not hurricanes, but weather events, rainfall that's enough to continue driving and sort of leaching this wetland carbon out of the landscape. And so one of the implications of that is that as we do that, we continue sort of pulsing this material out, is that it affects its reactivity. And so on the back end of the FAST process, we can also try to think about how long it takes for that carbon to turn over. And so I'm going to go just south of the Neuse River Estuary to the Newport River Estuary. Again, you can see this satellite image full of a lot of this wetland material. We did a separate study unrelated to the Neuse, but there what we were doing is we're trying to look at how the DOM quality through CDOM and all that influenced its carbon turnover. And we looked at aromatic carbon mineralization. So aromatic compounds of the part give rise to some of this color. And the important thing here is that we saw that aromatic mineralization rate increased with antecedent precipitation. Again, kind of a manifestation of the seesaw, moving that carbon out and then having it be reactive and turned over. So I kind of went back to 2018 and thought, okay, we've got this experiment. It turns out that there's this wetland connection that is very strong. What happens then if we try to estimate one of the outcomes of these pulse events and look at it in terms of how the estuary behaves with CO2 fluxes? So we estimated CO2 fluxes from the surface waters of the NRE and aggregated that here in this violin plot. And it's in balance except for most of the year, except for these particular events. And if you look at the antecedent precipitation, which is, I plotted this, it looks like I plotted this wrong, but you get the sense that the right around here is where all the water is coming through. And then look at this lingering effect. We saw this after Hurricane Matthew too, and what we hypothesized was that we have this kind of lingering, slow degradation of this carbon because it gets retained in this long residence time system. System gets saturated with carbon, and that stuff just gets worked on. It eventually, makes its way out. But you can also see that we can guesstimate that there's some fairly substantial carbon processing that happens. So just kind of wrapping up here, I think in terms of thinking about glimmer and fast processes, we can observe those happening, but then we want to be able to continue observing these systems afterwards to try to see what the slow, chronic, perhaps, effects are of these carbon fluxes. And so we kind of come up with this cartoon here that explains our thinking then. And if we look at this gradient of increasing flood frequency, what that's doing is it's driving more of the wetland source carbon into the system, and it's maintaining it as a CO2 source, especially when we have this sort of long residence time systems where these processes can be observed. And so just linking back here to that block diagram from Jim Bauer's paper. So I'll wrap up here by making a couple of points. I think these fast processes can create these long-term lingering effects. That's the main take-home message I would like you all to have here. The second one's also important, and it gets back to something that was mentioned by Erica yesterday during the report out. One of the first report outs is that we need to be, as a community of practice in these different types of scientific endeavors, we can't just go storm chasing and look at these events. We need to be able to know what the background is, to have the context to be able to understand how frequencies of these events are changing and what that means for coastal carbon cycling, for ecosystem health, for a whole host of things. I think this fluorescence method might be something that could be brought to bear to give us a little bit more information on these fast timescales. It's very easy to do these fluorescence measurements, and you get a good deal of information out of them. And then the last thing I'll note is that what we started working on, I'm in a Department of Marine, Earth, and Atmospheric Sciences, or Earth, Wind, and Fire, some people like to say. But the atmospheric people, the meteorologists, working with the State Climate Office, they have models of the historic Hurricane Matthew and Hurricane Florence they created for Department of Transportation to look at flooding. We're trying to work with those because they also have future models of those storms under different climate scenarios. And we're trying to work on looking at the responses, the resilience and resistance metrics for carbon and ecosystem processes using that modeling system. So I think a lot of these efforts are right in line with Glimmer, and be happy to talk more with folks about it later. And I'll leave it there. If you're interested in joining the Seesaw, please reach out. Thanks.
All right, awesome. Thank you so much for having me here today. My name is Sasha Kramer, and I'm currently a postdoc at MBARI. Today, I'm going to be talking to you a little bit about the types of data we might need to detect the impacts of wildfires on coastal ecosystems. And fortunately for me, the prior talks this whole week have really set it up really well for this talk. Both Douglas's talk on Monday and then Chris's talk just now, I think, are super relevant to the content that I'm going to present. So Douglas gave a great overview of some of the ways that we might expect a coastal wildfire to impact a coastal ecosystem. But I just wanted to start with this example. Let's say we have a coastal ecosystem where the phytoplankton community is mostly controlled by nutrients and light. If a wildfire starts in that region, this input of smoke and ash is going to impact both the availability of sunlight by decreasing the amount and the wavelengths of light that reach the surface ocean, and also impacts the nutrients. And so we have some suppositions about how that balance of decreased light and increased nutrients, perhaps also thinking maybe about if there are some inputs of things like heavy metals from wildfire ash, might impact the phytoplankton community. But we're really limited by the number of observations that we've been able to make of these type of impacts, especially in the ocean. There's a bit more work looking at things like coastal or high latitude lakes where there are wildfires happening, but the ocean is really not a place where we've looked at this very much. And part of the reason for that is that it's really difficult to mobilize a quick study in oceanography. And you all know this well, but when a wildfire starts, we don't have that lead time that we might even have with something like a hurricane where there can be some predictions. It just happens. So I'm going to give an example today from a case study during the Thomas Fire, which happened in Ventura and Santa Barbara counties in 2017 into 2018. So you can see here an example satellite image of the first day that the fire started. You can see how that wildfire smoke is going right over the Santa Barbara Channel, which is a highly productive ecosystem. We were really fortunate to have a serendipitously planned research cruise in the Santa Barbara Channel at the same time as this fire started. So we had an opportunity to take some samples from under the smoke plume. But you can see from this satellite image that that's the view that we have otherwise if we don't have an opportunity to go out in the water. This is what it looks like from above the smoke, and this is what it looks like on the ground. So I don't know about all of you who do remote sensing work, but this is not, to me, an ideal condition for remote sensing, or honestly for human life. So it was challenging conditions, but we were really excited to have this chance to go out and sample and see some of the things that we might find. So I'm showing now on the left a true color image of December 19th, 2017, one of the days that we were sampling in the channel. And then on the right is a chlorophyll image that we took from MODIS data. And I'm not trying to show this comparison as some kind of gotcha of like, oh, look, that satellite data was published even though there was wildfire smoke there and it wasn't even flagged. That is what happened, but it's really difficult to quality control all the satellite data that's being produced in the entire world every single day. And so there are inevitably going to be cases like this where we have atmospheric conditions that aren't ideal for producing satellite data, but those atmospheric conditions are not what people are looking for when they're flagging satellite data for atmospheric correction issues. So we have this map of what looks like chlorophyll concentrations, but I'm not so sure that that's actually what the chlorophyll looked like in the water. So fortunately, that little dot on the map is where we were in the Santa Barbara Channel on that day. So you can see some results that we have from the different measurements that we were taking. We were able to compare HPLC chlorophyll concentrations in green to some measurements of chlorophyll that were estimated from reflectance spectra measured with an in situ instrument below the smoke plume in red, and from MODIS above the smoke plume in blue. And the really interesting result here is that we ended up with both cases where we had an overestimation of chlorophyll and also an underestimation of chlorophyll concentrations. We were only able to put all of this data into context thanks to our time series data that we have in the Santa Barbara Channel. So what Chris was just talking about with having these sustained observations in these ecosystems where these extreme events might happen is so important. And if you saw Dante Capone's excellent poster earlier this week, he also used time series data from the Santa Cruz Wharf to look at the conditions during a wildfire. I think we really need to have these type of sustained observations when we can't get out in a ship and ground truth these type of measurements. And now the last slide I'm going to leave you with, you might think of wildfires, especially in the United States, as a West Coast issue, but here we are in Cape Cod, and we have wildfire smoke reaching the ocean here. So just keep that in mind as you're doing your remote sensing and your in-water studies. Thank you.
The next lightning talk will be by Enrico Sa from Louisiana State University, Coastal Biogeochemical Response to Flooding Events in the Northern Gulf of Mexico, from Ocean Color.
Yeah, thank you and hi everyone. So here what we are, first I would like to thank my co-authors, Bing-Ching Liu from University of Louisiana, Lafayette, and Ishan Joshi. And we also want to acknowledge funding from NASA So in this work, I'm going to focus on three events. Three estuaries. We have Galveston Bay, which is a estuary with one inlet, and it was impacted by Hurricane Harvey in August 2017. We also talk a bit about Hurricane Barry in July 2019 that impacted Mississippi Delta, which is a Delta front estuary, and Hurricane Michael in October 2018. This was a Category 5 hurricane that made landfall in October 2018. So this will essentially, what Joe alluded to earlier about fast processes. I'll just emphasize that all this work really needs to be monitored as fast processes. And the second thing was what Chris mentioned about long-term lingering effects. So both of them will be addressed here. So in the case of Hurricane Harvey in the Galveston Bay, we have, as you can see, we have the field CDOM absorption. And you can see the first one is just before the hurricane. Then August 31st, just after the hurricane, you see a huge pulse of that was transported into the that was transported into the coast. And then 1 month afterwards and 2 months after that. So we did some field work in this and then developed some algorithms for CDOM and backscattering. And you can see also particles that we derived from satellite data. One of the important things here is that here we use these satellite images together with field measurements of currents and tides to calculate the flux of material, carbon fluxes out of the bay. And you can see, if I expanded this, you can see the pluses denote the days and we didn't have the satellite images. So sometimes we had the blank here. So we could, thus we can, using multiple satellite sensors such as SPACE and Sentinel, we could probably constrain the carbon fluxes much better. So I wanted to just show here that field data is also very important to understand the link between satellite surface and the subsurface. And here you can see this is sediments in the bay 1 month after the hurricane. And you can see this was well mixed water. CDOM was very high, backscattering, suspended sediments. And you can see, for example, in this area, we have phytoplankton chlorophyll is very high, but decreasing fast with depth. And so that was suggests light limitation. But the other point that we note is that in this case, we have mixing, so productivity is faster than the mixing, essentially. So that's very interesting, something that we can use for, use satellite data, something that we have a large number of satellite images, and we could probably try to see what processes are taking place within the water column. And in this case, we are looking at phytoplankton dynamics in the river-dominated systems. In this case, it was Hurricane Barry in July 2019. And you can see that here we have chlorophyll. The top row is before the hurricane. The bottom is after the hurricane, 1 week after the hurricane. And you can see there is an increase in chlorophyll. But more important, you can also see that looking at the size fractions, micro phytoplankton, nano, and pico, we have an extension of the microplankton into the shelf, outer shelf. But more important also you can see is that pico phytoplankton here, the fraction of is very high, and that is because that is a Gulf Stream, the Wamco eddy. You can see this is a Wamco eddy, but after the hurricane, which passed over this area, the pico phytoplankton decreased tremendously, and you see an increase of nano phytoplankton. And finally, I just want to point out that what we have here is that Hurricane Michael, we looked at the high resolution within the estuary, and then we have this lingering effects of fluxes that extended for even greater than 20 days or so. And so the other point is that we also had a shelf-wide transport of CDOM DOC and nutrients that contributed to offshore red tides and coccolithophore blooms. So what factors lead to this phytoplankton species succession? Is it nutrients, turbulence, high CDOM DOC? So we see that strong hurricanes can have both shelf-wide impacts And therefore, we need to resolve physical drivers and biogeochemical response at multiple spatiotemporal scales. Thank you.
Thank you so much, Hiriko. With our next talk, we will continue the discussion on impact on phytoplankton dynamics. Adrian Marchetti from University of North Carolina will discuss influence of physical processes on phytoplankton community structure in the Galapagos archipelago.
Hello, everyone. So I just want to thank the session organizers for inviting me to be able to contribute to the work that we've been doing. So I'm also another member of the North Carolina contingency here, but rather than focusing on the Neuse River estuary, I'm going to be taking you to the Galapagos Islands in the middle of the Pacific. And just basically, I hate to disappoint Heather, but I do consider myself a biological oceanographer. So I want to make sure to give credit to a colleague of mine as well that's been working on this, Harvey Syme, who's also in my department. And basically, as a biological oceanographer, I'm very interested in what is controlling the abundance and distribution of phytoplankton communities. And so what I've learned through the work that we've been doing here and in other places is that I think we've actually already heard the quote that all science is physics and the rest is stamp collecting. And I'm kind of starting to believe that in a way. And so, again, with physics, we're looking at how physics can control light availability, nutrient availability, turbulence, aggregation, all these processes that shape the phytoplankton communities that we think we have to account for. So just to take you to the Galapagos here, essentially the Galapagos Marine Reserve is comprised of about 14 different islands, and it's a very unique oceanographic setting. You can see here just from this slide, it has this convergence of these major currents, including probably the most predominant one being the Cromwell or the equatorial undercurrent that provides that cold, nutrient-rich water that sort of upwells mostly on the western side, but also influences other parts of the archipelago. We also have this warm current coming from the north that's mainly oligotrophic, and so that intersects as well with the Humboldt current that comes from the south, and actually that's the current that the penguins basically take in order to get to the Galapagos, and fairly unique in that there are penguins at the equator there. As you may have known from the Galapagos, from a little-known scientist named Darwin, that it is a very diverse place. That's where he formed his theories of natural selection, looking at the land-based organisms. But we can see that actually also in the marine environment, it's a very biodiverse community there. And it's also impacted by ENSO events. So we've just gone through an El Nino, actually. So you can use it sort of as a window in terms of how we can see what the consequences are of a warming ocean. So we participated on 5 research cruises from 2014 to 2022 in the area, which actually incorporated the El Nino that we sampled in 2015, as well as just the recent La Nina event that was actually persistent for quite a long time from 2020 to 2022. And the major objectives we had is to look at and determine the environmental controls on plankton dynamics, as well as to understand the effects of these ENSO cycles on the marine environment. So this is actually just showing our base. This is the Galapagos Science Center, which is co-run by UNC and University of San Francisco, Quito. It was built in 2011. They're modern labs, and so if you're ever interested in doing work down there, just come talk to me, and I'd happy to tell you all about this incredible facility that we have on San Cristobal Island in the Galapagos. So again, my work is mostly on phytoplankton. So as we know, phytoplankton are fairly diverse. They come in all different shapes and sizes and have different traits associated with that diversity that become very important in relation to looking at how the environment shapes these communities. And in particular, in this area, it's really important because they actually, as we're concerned about here, they contribute to carbon cycles, but they also form the base of the food chain. And in places like the Galapagos, they play a major role as primary producers, which support all the charismatic organisms that people go to the Galapagos to be able to see. So in terms of our cruises here, as you can see, we actually were able to cover quite a few areas of the archipelago. All our stations are in red here. So essentially, we had 29 stations throughout the Galapagos. And basically what we try to do is they are sort of close to the islands because there were also other people on the boat looking at sea lions and iguanas that they would do more land-based work. So we couldn't go too far offshore because we had to coordinate with them. But we would try to go in about 100 meters of water so we can sort of remove ourselves from any sort of shallow embayment effects. Now, 15 of these stations we actually sampled throughout the water column. So we're actually able to get sort of characteristics throughout the euphotic zone. And then actually, I just want to point out station 14. I'll come back to this. This is a fairly unique station that we sampled within Darwin Bay, which is basically a crater that's been invaded by the sea. So it has a very unique community that live within that crater. So the ship that we use, this is the MV Sierra Negra. By no means is it a research vessel. So we've had to be very creative in how we do our sampling. So we're doing basically, this is Harvey here, doing Niskin sampling. And there actually is no room there's no lab in the boat. So we had to construct a garden shed, and this is Christina in the garden shed, where we did all our sampling. And so, I mean, I think, even though we had very primitive ways, we were able to actually measure a whole suite of different parameters that really gave us a sense about the phytoplankton communities. For a couple of years, we also had Scott Gifford, who's part of our department, and he's focused on bacteria. And so he actually published a paper recently looking at bacterial communities in the region and how they were influenced by El Nino. So just in terms of these cruises here, as you can see, this is looking at the index, the 1.2 index, which encompasses the Galapagos Islands. And you can see throughout these cruises, we have various levels of the anomaly, of the sea surface temperature anomaly. And most importantly, in 2015, you can see here is El Nino sampling here, as well as in 2022, we have sampling during the La Nina. So which incorporates a sort of variety of different environmental characteristics associated with these sampling time points. And so the bulk of my work I'm going to be showing you is mostly looking at community composition of the phytoplankton based off of molecular sequencing. We use 18S, the V4 region of that. And so this is a former student of mine, Erica Neve, who published a paper recently in 2020 comparing the communities between the El Nino and the 2016 event. And what I want to kind of highlight here is that she found that if you look at the communities and comparing the different years, we have sort of certain groups that dominate these, including dinoflagellates, chlorophytes, and diatoms. And then what we also found is that if you look at, so she did an MDS analysis, that if we look at where these different groups kind of lie, the dinoflagellates seem to be more spatially separated, so temporally separated between the years. So there was a distinct community of dinoflagellates in the El Nino versus the neutral year, whereas chlorophytes and diatoms seem to be more spatially separated according to the different sites that we actually sampled. And then lastly, what she also was able to kind of take away from her data is that water mass properties are really important in shaping these communities. And again, it's this convergence of all these different water masses that we think are really important in influencing the seed composition of the communities that then end up proliferating in the Galapagos. Then I had another postdoc here, Se-Hyun Jang, who is very good at looking at dinoflagellates. And he actually separated out, this is from a 2018 cruise, he separated out different dinoflagellates based off of their trophic modes, whether they be heterotrophic, autotrophic, or mixotrophic. And now a lot of that was based off taxonomy. And he found that the eastern region, so along the archipelago, as opposed to the EUC influenced region, which is mostly on the west side of Isabella Island, had very distinct protist communities. And in particular, these protist communities sort of fell out in relation to the environmental characteristics that were separating out these two regions of the upwelling dominated versus the eastern regions. In addition to that, we also were able to opportunistically sample a red tide. And this is actually one of the very first instances I've actually encountered where the water was red. And so it was an actual bloom of this dinoflagellate here, Scripsiella, as well as a heterotrophic dinoflagellate, Polycricros, that was feeding on that Scripsiella. So again, a very opportunistic sampling that we were able to do just because we were there. Otherwise, nobody would have known about it at all. I want to then just sort of focus on some of Priska's work here. Priska actually just completed her master's in my lab, and this is looking at all the different years that we've sampled. And so I just want to boil this down a little bit. And what you can see here, this is actually a WGCNA plot, and Harriet actually showed one in her talk a few days ago. So I don't think I have to sort of, well, I'll describe it very briefly here. What we can do with these WGCNAs, we can start to sort of cluster organisms based on environmental characteristics. And so when Priska did this, what she actually found was that there is this module here in this blue, so this group of organisms that were highly correlated to lower temperatures and increased nutrients. And so we considered that cluster associated with an upwelling module. So these organisms tend to do really well during those upwelling events. And as you can see here, looking at across the different years, they spatially separate out where we see, in fact, those stations that we sampled upwelling, we tend to have more of these groups associated with them. But another thing that's to point out is that they don't just necessarily confine to certain taxonomic groups. So we often talk about how diatoms are really good at high nutrients, but we also found that there were chlorophytes, certain genera of chlorophytes as well as dinoflagellates that seem to group together during these particular environmental conditions. And then we have this cluster here in turquoise where we have warmer temperatures and lower nutrients. And you can see here that this group here was mainly confined temporally and really did well, or really sort of was present during that El Nino event. So again, associated with those warmer, more oligotrophic conditions. And we're very mixed communities. So I think our perception of functional groups of phytoplankton being where, diatoms dominate one region, dinoflagellates another, we have to sort of rethink about that because there are sort of mixtures in the community, and it's probably not as simple as saying that they separate so easily. I also want to show some of Margarita's work here, and Margarita is a current master's student in my lab. And so what she did was, again, knowing that we have this physical environment that really we think is shaping the community here, she basically is plotting the potential density at the surface versus the change in temperature at the interface, so it's sort of at the subthermocline, so which is more, we think, essentially this is a proxy for looking at temperature, so higher density water with cooler temperatures. And this is sort of a proxy for mixed layer dynamics. Is there, an upwelling? Is it basically very little mixing going on between the surface and the deep versus where you have a stratification where there's a lot of mixing and a distinct mixed layer? And, what she found, again, was looking at these communities. And so she's plotting here, these larger circles represent more chlorophyll. And basically the color is an indication of whether it's in the large size fraction of the phytoplankton versus the smaller size fractions. She can separate out spatially these different groups so we have this West Fernandina station, this outer east station, which is sort of over here. And then here's that Genovesa station, which is that in Darwin Bay, that very unique community that we're seeing. So again, sort of highlighting how we're seeing these distinct phytoplankton communities associated with these different environments in both space and time. So I wanted to sort of summarize this here, and I want to get into a little bit of future work that we have planned. So microbial diversity is very high in the Galapagos, and we think it's very influenced by changes in this water mass properties. And in the El Nino, we definitely see this warmer, sort of reduced nutrient sort of environment where we had reduced biomass and productivity. During the La Nina, I didn't really get into this in specifically, but what was really interesting there is that we found that even though we had cooler waters, there was actually this influence of more fresher waters along the surface. And we think that may have influenced stratification and prevented a lot of those nutrients from the equatorial undercurrent actually making their way up into the euphotic zone. So we actually had less biomass and productivity than what was anticipated. And then lastly, just to kind of highlight here, plankton productivity and community composition are shaped by physical processes in varying spatial and temporal scales. But then the big question is how? What is the predominant factors in the environment that are influencing these organisms? And what we think is happening here is that essentially, if you think about functional traits or of organisms, you have these very mixed communities here, and they can have varying taxonomic compositions, but you may have redundancy in that taxonomy in the sense that what probably the environment is selecting on is functional traits. And so these sort of represent functional traits, and it can be in relation to light acquisition, nutrient acquisition, and perhaps then that could be a very important factor in structuring the community. And so in relation to nutrient acquisition, for example, we can have situations where we have those that do really well under high nutrient conditions, so we consider those of VMAX specialists, versus those that do well under lower nutrient conditions, as well as we learn a lot about during this workshop, nutrient storage can actually play a distinct role. So we think that these characteristics or traits can be very important in shaping these phytoplankton communities. And with that in mind, we actually want to, we have a new NSF project that we've got funded here. And actually, Cynthia had mentioned this new program, Biodiversity on a Changing Planet. And so that's the program we actually recently got funded by. So it's Harvey and myself, Monique Messi from MBARI, and Diego Figueroa, who's a zooplankton ecologist from University of Texas, Rio Grande. And a key component of this program, if you're interested in it, is that you have to sort of address three main themes. And the first is functional diversity, the second is biodiversity dynamics, and the third is a changing planet. And so the work that we've been doing in the Galapagos sort of hit on all these themes quite well, hence I think the reason why I like to think why we were funded. But what we're doing here now is we're actually now trying to really get at how physical modes influence the nutrient inputs into the euphotic zone and shape these plankton communities. And so to do so, we're actually going to set up, we have two mooring sites, one being right outside the GSC to start off with, and we're going to have a wire walker so we can look at water column dynamics. We actually have a cruise lined up, hopefully sometime in 2025. We're going to be doing some culturing experiments. You know, as a PhD student, my advisor always told me, keep 1 foot in the lab and 1 foot in the field, and I've tried to do that throughout my career. So we're going to be doing culturing experiments to look at these nutrient uptake kinetics, and then as well as take advantage of the enhanced capabilities of these new array of hyperspectral sensors like PACE and Glimmer to really help us fill in the sort of the holes of our sampling. And then lastly, do some modeling. And so just to finish off here, I'd like to talk to you a little about what we've been able to characterize in relation to these physical processes. And the first one, as I've mentioned, is this equatorial upwelling. And so we think here the time scales represent weeks to months in terms of this pattern of upwelling that we see. And this is looking at surface temperature as a function of depth here. And you can see that this is actually on the west side here, off of the coast of Isabella, so where there's predominantly upwelling. But those trade winds die down every January to March, which actually sort of increases the temperature, but then you have the turn on of the upwelling. So it has this very sort of rhythmic pattern based on sort of longer time scales that we think can result in persistent injections of nutrients. Secondly, we've been able to identify island wakes as a very important characteristic to defining or to enhancing nutrient uptake. So if we look here off San Cristobal Island, so this is sort of the transect across here, we can see these periods of higher productivity. So this is just showing modeled productivity from a satellite product. We see these spurts of higher productivity occurring, which we think, again, are associated with island wakes, as opposed to, say, down here off the coast of Isabella, where we have that more consistent upwelling. So island wakes, we think, are in shorter time scales, days to weeks, and have high nutrients, but very sporadic injections that could then be important in defining the phytoplankton communities. And then lastly, we look at internal tides. And internal tides here, what we did here is at one particular station, we basically just did a series of CTDs over a few hours, and we can see the thermocline jumping up and down about 30 meters. And with that, we can see the sort of the concentration versus sort of the spread out of the chlorophyll associated with that internal tide. Again, so we think that the time scales here are looking at hours to days perhaps, and then you may have sort of concentrations of periodic injections. So all these different modes of nutrient input, again, we think will preferentially basically define the nutrient acquisition strategies and thus define the phytoplankton communities. And so just to finish off here, if I can, so essentially, we know there are challenges associated with satellite imagery, especially in the Galapagos. There is a lot of cloud cover, but I mean, there are, you know, windows of opportunity here to actually get at really good images. And so this is a really nice image from PACE that was from May 24th, so a few weeks ago, where you can see, again, probably these island wakes having a really important influence on the increases in chlorophyll. And so just to finish up here, I also just wanted to say how climate change, if we're going to learn how the effects of climate change, this is a really interesting place to do it. Not only is it, again, affected by El Nino here, but you can see that there's been a change in both intensity and frequency of El Nino events, which could have important impacts. So we can use the Galapagos as a natural laboratory to study this. In addition to that, what's really interesting, there's these papers that recently came out talking about how with the warming oceans, we're going to have migration poleward. But if you're in the Galapagos and you're a phytoplankton, you can't move north or south because you're entering warmer waters. Okay, so you really have to either adapt or you basically won't survive in warming. And lastly, there was a recent paper that came out from more satellite data that suggests that there are certain parts of the Galapagos that are actually cooling. And this was a really study by Chris Karnauskas, that over the last 40 years, the EUC has been intensifying. And so there's actually been a change of about 0.5 degrees Celsius on the western side, whereas the rest of the archipelago has been shown to warm. So this is a really interesting place to look at how that variation in temperature will play out on the marine ecosystem. And then, so, you know, I think that the way to do this is so we should be doing some long-term monitoring of this region. And by using a variety of approaches, I think we're going to get at hopefully a better understanding of what's going on. So with that, there's a whole group of people that were involved in this. And so I'd like to thank them as well as the funding agencies, which have mostly been internal to UNC, but just recently also NSF. Thanks for your time.
That was fantastic, Adrian. Thank you so much. Our final talk for the session before our break is going to be a lightning talk by Broward Johnson, University of New Hampshire, how spatial and temporal resolution need to be matched.
Thank you so much. So yes, my name is Broward Johnson, and I'm at the University of New Hampshire. And I'm going to start with what I'm going to try to do is kind of explain or discuss why should we care about these fast processes? And this is coming out of being interested in episodic events, very short time scales, and how things are connected over 10 years. And many times people are, yeah, but you know, if we average everything, it's going to go away. So we don't care. And the reason, I just want to start with the concept about climate models. So this is just an example of how the resolution of climate models have changed over the years. So this is the first assessment report, second assessment report, third assessment report. We need to figure out how to name things because we don't know how to do the fourth assessment report. So we start with assessment report 4. So this is the IPCC report. And you can see how the resolution has increased pretty dramatically. Now we're coming down to climate models that are about 10 kilometers. And the reason why we do this is because we want to see we want to be able to resolve certain processes better and better. To the left here, we have an example of a climate model with a 1 degree resolution. It's sea surface temperature and chlorophyll. And to the right, we have sea surface temperature from a satellite with about 4 kilometer resolution. And you can see that the large scale patterns looks very similar. But when you start to look at the coast, you can the coarse model misses all the upwelling. And that's kind of where people live. So we kind of want to be able to pick that up better. So in that sense, we need to, that's why we need these high resolution models. And the problem with this is that, and this is another example of this. Here we have, sorry, Google, the Google system crashed my image, my presentation. But here you see the white squares here is a 1 degree climate model, which is normally what we're using for biogeochemical analysis, etc. The large box is a 1/4 degree resolution model, and the little strip there is Amala Mahadevan's 1 kilometer resolution. So you can see how much more of variability you get in the high resolution model. But not only do you get that, you also get much more resolution and much more processes in the vertical. So this is the same box, but we're just looking at how it looks at depth. And you can see you have these very fine elements going up and down, a lot of submesoscale processes, which makes it much more complicated, much more interesting. We also know that time and space are connected. So if a spatial resolution, a process with a certain spatial resolution, or with a smaller spatial resolution is also working on shorter temporal resolutions. So therefore, we need to kind of combine those two questions. And that's where it gets complicated, because a model is a simplification that only works for a system where we answer a specific question. And for ocean models, or for GCMs in general, that means that if we're looking at a certain timescale where we develop the model, everything works really well. But if we go too far into the short timescales, we just get numerical noise. On the other hand, if we go too far into the long timescales, we just get a model that cycles. Basically, if we take a model and we spin it up and it works really, really well over a year, and then we start running it over decades, we're not going to be able to see what's happening over the long terms. So that means that we really need to be able to understand the processes on the short timescales. And when we are now coming to these climate models, these BGCMs that have really, really high resolutions, we need to be able to understand and how to model the biological processes on very short timescales, including dial timescales. And this is where we have the big major challenge. So this is what we want to work with Glimmer, for example. And some of the challenges are temporal strategies, like storage, migration, we've been talking about, circadian rhythm, trophic interactions, non-steady state, dial traits, and biogeography. And when it comes to trophic interactions, this is a great paper by François Barret out of UW, where it shows how just the dial cycle of phytoplankton and grazers can make complete different stories in how they interact. So this is a very, very important part to bring into the models. Going back to what Joe showed with these studies we did in using Gochi data, what we see here is, and I should say this figure that Joe has already shown has about 100,000 data points for each of these lines. So what we see here is that we get a very different productivity in the morning and the evening. This is not something you would get from a model as they are set up today. Why do we get that? We don't know. But this is something we want to work on, and this is where these resolutions are very important. This is just another study that got the same results from Garcia Robledo. And that is basically what I want to talk about, that this is important. We should care. These are some scientific questions that a group of us have been discussing about what should we do and what do we need to think more about to be able to use these high temporal resolutions in climate models, in biogeochemical models, and how to kind of bring these communities together. Thank you.
Thank you, everyone, for coming back. We're going to continue with the panel discussion. We have all our speakers there, so we can open it to the floor for questions. Also, you see on the slide, we have a Glimmer survey. We would like you to use this QR code, and whenever you find an opportunity, please provide feedback on how you think you could use measurements from this new geostationary ocean color instrument, and more importantly, provide some information on what you would like to see. What products that we're going to be developing you feel would be particularly useful for your research. And if you don't see any products that you feel you would like to use in your research to help with your models, your analysis, remote sensing observations, please provide this information so that science teams, future groups can focus on developing some of these products. So now we can continue with a panel discussion. Any questions for any of the talks we had this morning?
Thank you. First off, it's just an amazing set of talks. So congrats to the conveners and to everybody who spoke. So a question to Joe about Glimmer. It's really cool that the satellite will be flexible enough to focus in on particular events, but I'm curious, how will those events be prioritized, and is there an impact on long-term observing on other parts of the footprint there if it's kind of optically focused on some specific event?
Yeah, I mean, I know what we proposed, and since then, we understand more about operations and how this would actually happen. We have, I guess, kind of competing interests of maintaining a mission continuity and doing the same thing to, wow, there's an amazing event of societal importance or scientific importance. We should sacrifice that time and come over. So we've talked about, perhaps a web interface where very quickly Antonio and I, Maria and other science team members could have a quick conference call and talk it over, thumbs up or thumbs down. But there will be requirements that we're going to need to develop over there. We're still a few years away from launch, so that's in the background. We might, we realize we might need more money for operations people because they've got to reprogram everything and, send messages to the satellite and whatnot. So, TBD on a lot of it, but it's a real high interest of me personally to execute that.
Hi, Dante Capone from Scripps. Thank you all for the really interesting talks. And this was mainly a question for Chris. So given that these extreme events might occur in unpredictable regions, in the absence of time series monitoring, what recommendations do you have for developing a structured in situ sampling plan and carrying that into the future after the extreme event might occur?
That's a great question, and has sort of been the way we've operated. So we see an event coming and we mobilize to try to get out there and sample the system before the event, if we can do it. And then after the event, so imagine you can look at the return to baseline conditions as best as you can. That actually brings up a point. So the Seesaw had a small meeting at the surf meeting last fall. And we were chatting, and Nathan came up with the idea, I'm gonna put him on the spot, came up with the idea to reach out to DOD or other entities that can get into denied areas and have expertise in doing sampling or going into dangerous environments. Because a lot of times that's what, you were saying, like, how do you continue on after that? Well, there's a lot of gaps, several days, if not weeks, after the event, because conditions have to be safe enough that we can go observe those systems. So that's why Glimmer is important, but it's also, trying to build those partnerships and that network to be able to do exactly what you're asking.
This is Blake Schaefer from the EPA, and I wanted to respond on the event sampling that Joe was answering. I can't speak to the operations side of it, but we did do a study because, as you can imagine, we're really interested in event monitoring in between what Glimmer will be doing. And we published a paper in Marine Pollution Bulletin that the sampling rate is actually really quick. And we did a simulation, like if you wanted to replicate a swath from PACE or something like that for an event, it's actually not that much time for Glimmer to do the acquisition. So all that's kind of been looked at, and hopefully it helps with the operations.
Can you hear me? Yes. Suzanne Craig, NASA Goddard. So full disclosure, I was one of the conveners of this, so I bit of an insider question. What are you going to do about atmospheric correction? because it doesn't really work at the coast for lots of reasons, algorithmic reasons, and mainly the fact that we don't get the atmospheric models right in order to remove them accurately. So do you have a plan?
Yeah, we have a present plan. We're going to do as best job that we possibly can. And let me turn that one over to Antonio, who may have a better, better job.
Yes.
Answering.
So our lead for the atmospheric correction is are Brian Franz and Amir Ibrahim, and they're the leads on the PACE atmospheric correction. And so, as Suzanne said, that is definitely a concern. I know that Amir has, for PACE, has implemented pseudo-spherical shell models. So that basically means they're taking into account the full, the Earth as a sphere as opposed to, you know, a 2D surface. So that's an important step. Also, they've implemented, starting to implement for PACE, this multi-band atmospheric correction. So traditionally, for ocean color, the OBPG at Goddard has used 2 near-infrared bands. With this MBAC, they're using more than 2, and that should help better constrain the aerosol model, which is the most challenging part, and that's one area. The other aspect are the absorbing gases, and so we hope to be able to, as Joe mentioned, the PRISM provides high spectral and sampling and resolution in the lower wavelengths. We hope to be able to retrieve NO2, which is one problem. So that's how we hope to mitigate, and also we can detect absorbing aerosols, which are particularly problematic. I don't have a solution for that. I'm not an atmospheric correction expert. Another thing I would add is that there's an IOCCG, International Ocean Color Coordinating Group, that they're convening a team of scientists to study, to address a number of topics related to geostationary ocean color, and that's a primary one. That hasn't really gotten started. They're just actually requesting for applicants right now. You want to follow up? Yeah.
And to add to that, also, there's going to be, there are already atmospheric sensors on geostationary platforms such as TEMPO for NASA and other, across different other agencies. And this information could also be used both for aerosols as well as atmospheric trace gases in coastal areas.
So I'm just going to be the troublemaker in the room and say we should change the paradigm. Don't do straightforward atmospheric correction as we have done. There are other ways to do it. And I'd love to chat more with you about that. I won't go down a big rabbit hole right now because this is not an ocean optics conference.
Yeah, I mean, since the history of ocean color, atmospheric correction should be a growth industry, and yet it kind of goes through these fits and starts and very, very small incremental improvements. So I'm in agreement. We need it.
It's incremental agreement because it's, at least for these very complex and very biogeochemically important coastal regions, not the right approach. But I'm just going to be a troublemaker. But I'd love to chat with you all.
But let's find the right approach.
Other time, yeah.
You know, let's find the right approach. We've got a couple of years to do it.
Yeah, I mean, PACE is also, as you know, I mean, being part of PACE Project Science also, you know how we're looking at the coastal environment is also important for PACE. So there will be, you know, we have at least 3 years. So it's probably 3.5, so maybe a little more. So we have time. Time is on our side.
Hi, this is Sherry Flogi, Wake Forest University. I have a question for Chris and Nathan. I'm curious about your increased C2M releases into the Neuse River estuary and if you've seen an association with reduced productivity, similar to what's been observed in the Gulf of Maine. So is productivity being measured, and do you see any inverse relationships with C2M?
I'll answer that. I don't know. You know, with the CDOM also comes a lot of you know, nutrients too. So yeah, there's the light limitation thing, but there's also the nutrient loading thing, and I don't know which one wins. That's a really interesting question. And we have the data. I just haven't looked to see.
That was a great answer. there's a lot of work we can still do.
Hello, Luai from Hui. I kind of have a half-completed thought / question. This session really made me think about the research cruises that we go on, and they're quite sporadic. We go to a station, take a sample, move on. Do we really need to start thinking more about quote-unquote geostationary cruises or Lagrangian cruises? And if not, how can we kind of connect these cruises that cover a lot of distance but not a lot of temporal resolution with the satellite data that you guys are thinking about, and then so on?
Yeah, let me take this one, and other folks can follow up. Absolutely, yes. Absolutely, yes. I mean, so I lead a couple of cruises, the COA cruise and for NOAA, and we have stayed on station with remote, even though it's for carbonate parameters. I've convinced NOAA to allow us to drift with a drifter. We've done the same thing in Korea under a so-called Chorus cruise in 2016. But absolutely, we need to understand those diurnal dynamics, and there are different ways to do it. With a big ship, it's a little more difficult, but if you are actually following a passive drogue, then perhaps you can. But absolutely, these are important time scales that aren't well resolved. I don't know if anyone else wants to talk about that. Yeah, go ahead.
It's a big issue, and I think even like in the context of the program that we've been trying to develop called BioGeoscapes, and we've modeled it under the GeoTraces sort of format, but that's very sort of survey-based. And so we understand that, a lot of things that are happening in the ocean are essentially happening on, different time scales, diurnal patterns of organisms and vertical migration and things like that. So I think we have to really sort of think about leveraging, time series data sets, observatories of like coastal study areas, moorings, like as we're doing with the wire walker, because we're not getting the full picture by just doing snapshots of large areas. So I agree with your comment.
I want to add that we have a project in Hawaii where we are using sail drones, and this comes from private funds. So there are, you can also leverage other type of interest from different communities. And we have been collecting data from all islands using sail drones 3 times a year. And also we will be able to sample an area after the fire in Lahaina in Maui. So just putting that there, that there is other platforms that we can leverage too.
Yeah, I think I agree with everything everyone is saying here. I feel like we go on these cruises and you are trying to compare these different areas, but you're actually only seeing them in snapshots in time. So you're saying, oh, this area is more productive than this area, but like it could have been an event-based thing. But I think to increase our temporal resolution, we can really start to look at more creative options like the sail drones, and getting more autonomous type measurements on different kinds of boats than we typically think of, like not just research cruises, but we can also start using ferries and like things that regularly go on the same tracks over and over and over again multiple times a day. That would be a huge increase in temporal sampling.
So I just want to add that a lot of coastal waters, estuaries, they have strong spatio-temporal gradients. And one of the things that, just from our work, for example, we can do a transect, from, say, the upper bay to the lower, to the coastal waters. But it takes about, 6 hours or something, to do that transect. And I think that is perfect. Don't need to stay in one place. Since Gleamer, for example, is taking snapshots every hour or so, as we go along this transects, we are going to get the samples that correspond to different time frames. And therefore, we really don't need to stick in one place and do that type of, not always, but generally it can help, because by staying in one place, especially in a coastal system, you are not looking at the spatio-temporal differences that are there actually. You need to consider also that doing a transect is also good, because, and that's especially the case with Gleamer, unlike Maurice and Pace, because they just pass at certain hour, at 1:30 or something like that. And so we are making those measurements there may be one station, then there is a plus and minus 4 hours around that satellite path. But with Glimmer, you're going to get, you're doing the whole transect over a large area and over different time frames. You're going to get data that is going to be sort of at the same time as Glimmer paths, because it is a sequential time. So I think that's another option to do some sampling also, which is optimal for coastal waters.
Is this a format for debate as well up here? Because I would just like to counter my good friend Enrico with just a point. I mean, part of the reason, and we've done this right in your backyard too, is that let's set a drogue in and trace the drogue, because we want that particular water mass, and so with this high gradients, we may move across. So everything he's saying is absolutely correct, but at the same time, I think there might be reasons for these in situ studies where we're just tracing a water mass over the course of the day.
I'm not saying that.
Yeah, I know.
Combining both of them is just good enough also.
One thing I wanted to, sorry, go ahead.
I have a question online, but did you want to complete that thought? Go ahead.
I was just going to follow up on.
Go ahead. You go first, then I'll do online.
Can you hear me? Okay. Julia Moriarty, CU Boulder. I have a question about, so there's a lot of different types of events. They all have some different characteristics in their response. And one of the things I was intrigued by in Chris's talk is this resistance and resilience metric. That didn't seem, usually you see those words in association with communities, human communities, but not so much like the biogeochemical or ecological response. And I was wondering if you could speak more about that as like a framework for uniting some of these different responses to some of these different extreme events or fast processes. What like fast processes might bring to that.
Can I pass this over to a co-author on that paper?
Yeah.
Sounds great.
So the way those terms, resistance and resilience, were used in Patrick's paper that I helped out with, we were measuring resistance as the deviation from the mean for a ecosystem property, say productivity, divided by the strength of the force supplied. So maybe it was rainfall, precipitation, or wind speed during these hurricanes. So that's how we defined it. And the resilience was how long it took to return to the mean after the event. So I mean, it's a great paper. I encourage everybody to read it. But yeah, so that's how we were using it in that context. Does that answer the question? Yeah.
I'd also just quickly follow up and say that it's a nice paper because it gives rationale for having observational programs that aren't, oriented specifically around an event, because you need to know that return to baseline.
Okay, we have a question online from Bob Anderson. This question is for Joe. If I remember correctly, there was a proposal about 20 years ago for a geostationary satellite with a hyperspectral sensor, not selected for a mission. I think Janet Campbell was involved. Do I remember correctly? If so, then how does Glimmer compare with what was proposed then?
Yeah, so Janet was involved in that. I believe that was also a NOAA sensor, and I don't know if Janet's on. She may be online. She can pipe in, but I can't remember if it was hyperspectral or not, to tell you the truth. But yeah, and I don't remember much about the instrument because I'm a young man, and 20 years ago was a long time ago.
Yeah,
I
actually do remember that, but I don't remember any details.
Yeah.
I'm sorry, Bob.
I don't think it was hyperspectral.
Yeah, I don't either.
It was close.
Yeah.
If I remember correctly, Janet and her colleagues at JPL had proposed something called COCO.
Yeah, COCO.
And it was was a filter wheel system with a mini band, something like 50 or something. I don't remember the number, but it was a high number. It wasn't quite hyperspectral. And I don't know if they made it to a proposal. They did a white paper for the first decadal or science decadal survey, and that kind of led to the GeoCape mission pre-formulation activities.
So there's a direct link, that effort to.
Another question online, if we don't have any in the audience right now. Okay, this is from Ryan Vandermeulen. To everyone, does anyone have thoughts on how we better engage with our potential stakeholders now so that when Glimmer launches, people are ready to actively use it to study oil spills, improve HAB bulletins, and other?
I'll jump right in. I'm actually doing quite a bit of stakeholder engagement on a number of fronts and going out, talking, showing like what the data can do, and specifically trying to find people's pain points. What do they need? They want, fisheries, they want clean water, they're worried about harmful algal blooms, these types of things. You go and you talk to them about how Glimmer and these monitoring programs can help them out and try to make it in terms so they can understand. I have talked a lot about fluorescence and usually put the room to sleep, so it's also a challenge to do. You got to brush up on your science communication skills, but absolutely going out and just engaging, just talking to those communities is the best way to try to build those networks.
If I could rephrase Ryan's question slightly to this audience. I've talked to a number of you, and some of you have suggested, I'd really like to use satellite data, even current stuff, but I don't know how, or I need help. So this came partly from Nathan and I last night too. How does that work for you? How can our community help you get over that hurdle so you can use it for your papers and your students?
And I would urge you to fill out that survey because some of the questions have to do with how for you would be easier to get access to this data, what methods you use, what data files you would like to have, how you would like to learn about Glimmer using user guides, using web pages, there are many options there for you to choose, and then you can provide your own ideas as well. So really this is important for us to plan this engagement of the data user community and make sure that everybody will have access to this data and will be able to use for any kind of applications. Modeling, integration with field observations, integration with other satellite data sets.
And Ryan, I would say that one of the things that we need to do right away is to get your group and the Glimmer Science team talking about a strategy to do exactly this.
Just to address Blake's comment, just one thing for you all to, if you're interested in pursuing this and you're not familiar with using remote sensing data or ocean color remote sensing data, the PACE mission and the Ocean Biology Processing Group is putting out some videos, some webinars. They're doing CDAS training, which is a software tool to work with ocean color satellite data, distributed by our group. So look at those. There's going to be a training session in ocean optics. We're looking forward to having a training session at other conferences, some online things, as well as there's a hackathon that's happening, although it's closed now, this hackathon coming up in August, but the material will be available online. So, and we would definitely like your input in developing future training opportunities, training sessions, and where, what would be convenient, like OCB, is that a convenient venue, either the day before or the day after, if Heather and May are willing to accommodate us. I know they would, because they're incredible. So, more questions or comments?
Can I make a comment about the increasing accessibility to satellite data? I feel like there's a lot of professors and faculty in the room, and I don't know how many universities offer something like a satellite data processing or remote sensing course, but I think a lot of students would be really interested in taking something like that. Like, it could be formatted as something similar to a GIS course. I would definitely have taken that class if it existed.
So I just wanted to make a quick comment on that I've actually tried to incorporate that in my biological oceanography lab course.
in our, I would say, even lower level, we have to start having some coding classes as undergraduates, because they just don't have the capacity to even use it if you show them, right? So, sorry.
Okay, so so Jerry Wiggert, University of Southern Miss. The diurnal cycling, Nathan, that you showed for a motile organism, there's also physical mechanisms that drive sort of a diurnal cycling, a mixed layer or whatever, that then affects the phytoplankton community. And this is, I guess, maybe more of a comment, but maybe you guys can chime back in. It's really exciting to me that we're going to get to a state with the observations where you can start to resolve that process a bit and understand from the ocean color side what that looks like. Because, for an example, in the Arabian Sea in the northeast monsoon in the wintertime, you've got this cold air mass coming down that's trying to convectively mix, but then during the day you warm up. So you get like this mixed layer that goes from about 10 meters to 90 meters. And so it kind of regulates a bloom that's trying to happen. But in the satellite data we've had in the past, all of that's aliased into a longer term perspective. And I really am interested to see if you observe that, resolving over a daily cycle, what does that look like? Because I don't really have a sense for that.
In our estuaries, the stratification is predominantly driven by salinity. So the diel-thermal stratification that you're talking about, it does exist, but it really just works in the upper mixed layer. And it does seem, if you look at the traces of the depth of the, if you just look at temperature profiles rather than density, you can see the diel-thermal stratification really clearly. And the descent in the evening of the flagellates does seem like it's kind of like they're following the pattern of the deteriorating or the cooling and convection associated with the deepening diel mix layer. Yeah, I think there's something to that. Yeah, that's the observation, but I really haven't looked into it very carefully.
So one of the things is, of course, if you're talking about Arabian Sea, Lima is not going to give you that data actually. So I think alternatively is right now we have a lot of other polar orbiting satellites like PACE and these. The other satellites are there too, ocean colors from the Europeans, a Sentinel. So I feel that one way to sort of address, and they actually pass over this region different times, and maybe one can try to see certain trends can be observed over like 3 or 4 hours, because that's a time frame when any of these satellites, are sort of passing to a certain age.
Yeah, well, I should have prefaced, I recognize Glimmer would not see this, but there must be other locations where a physical mechanism like that is in place that Glimmer could be tasked to look at. Just as I don't know how that looks in ocean color, but it would be really interesting to see what it looks like. Because like we were talking, you showed the power sector and that Nyquist frequency where we can't resolve it below the sensing capacity. And that's been true in terms of that bloom dynamic so far.
Also, I would like to add Glimmer will be one satellite which will give you, especially in the Gulf of Mexico, about 6 passes. I think we should still consider, because we are talking about multi-temporal spatial scales too, because 300 meters, you can go out into the Gulf of Mexico. You may not need the large, the high resolution. So you could still add the other satellites that are still going to be there. I think so. Using multiple satellites data is really another option that should be considered in addition to.
Well, follow up to that, is there a coordination element there that can be facilitated? Or is it, in terms of all these various data streams, how we can manage to do that so it's simple for a user to get in and sort of access them?
CDAS at NASA is having, CDAS, you can process different satellite systems, right? You have Sentinel and other satellites. So
Sentinel-3, I don't think Sentinel-2.
yeah, Sentinel, but these are, yeah. At least we have 3 satellites.
But I think Jerry, you're trying to get to more of one-stop shopping for, overpasses at different times, and I'm not sure that exists.
It sounds like it's a proposal it's a proposal idea for somebody, not me, or maybe it's you.
All right, to avoid shortchanging our small group discussion time, I'm going to ask that we thank our amazing speakers one last time, because that was an incredible session. Really thought-provoking.
Okay, the things that I forget to say, my co-conspirators, please wave their arms, or else come right up here and take the mic away. But, first of all, thank you very much for hanging in there on the last day. Well over 80 people. It's a really high percentage. It's so beautiful out. This has just been a gorgeous week for weather. I'm kind of honored that everyone hung in there for the last session. It was really useful for us. We've got some great advice and ideas as we go forward. So thank you. I want to thank all the organizers here. Heather just does, and Ed L. do a great job year after year, and it's just been amazing how they've been able to keep this going for so long. Thanks a lot to the folks that helped organize this specific session. Some of them didn't do talks, but worked really hard on this, Maria, Antonio, and Blake in particular. And so a couple of things, if you want to follow along and stay involved. First of all, just reach out to any one of us. We answer emails if you have any questions about Glimmer or the status of the mission. Feel free. We are proposing to have monthly webinars that have to do with observational modeling capacities, sampling strategies, perhaps things relevant to the Glimmer mission as well. And we're talking in the early phases about having that organized through OCB. So stay tuned to that, read that newsletter and whatnot. And so those will be virtual meetings. Aurora and Maria are contacts on that, but stay tuned as well. So what else have I forgotten, friends? Okay, just thanks. All this stuff, any individual table may not have meant much to you in the half hour we're out there, but I think if we aggregate this and kind of see the ideas of the community coming through, it's going to make a big difference to the way that we propose our science work and our science teams. Again, please reach out if there are any questions, and that's it. Yeah, Blake, go ahead. Yeah. So again, we're trying to spin up either an article, maybe it'll just be an article, or maybe it will be a special issue that could highlight some of your work. And again, we will try and keep you informed on that through the OCB newsletter. Is that okay? And okay, kind of on the fly. There has been some work reaching out to publications. There's been some interest expressed. And we'll let you know how that goes. But, I know from talking to you, you are studying processes that are right in our wheelhouse. And we'd like to know more of this. This might be an opportunity to, consolidate some of that work into one special issue. What else? Okay, I guess that's it. Anything else, Heather? Oh, yeah. Can I get that right here? Woohoo! Yeah. Yeah, please fill this out. So we have, I told you that our applications work is is growing really rapidly, but it's not well funded. We may have an opportunity to get more funding for applications type work if we show more community interest. Kind of like a chicken and egg with NASA right now, or with our particular program. They want us to do all the work, and then maybe a little bit of funding will come in. So we have to do this incrementally. And if we can show interest in that, please help us out there. Right now, I think that's all. All right. Thanks a lot, folks.